let's go over cell division. So we're going to talk about mitosis and meiosis. And before we dive into that, it's important to review vocab and prior central ideas so that we can have the best understanding going into this. So there is just a visual representation of chromatin and chromosomes when we're talking about that. And then it's really important to review the cell cycle. So as we know, the cell cycle mainly consists of interphase. So interphase involves G1 or gap 1, S phase, also called synthesis, and G2. So during those gap phases, the main function is growth, and then during synthesis, there is replication. And after that, that small blue sliver is M phase, which we are going to focus on today. So M phase consists of prophase, metaphase, anaphase, telophase, and cytokinesis, which are part of mitosis. So mitosis is cell division that occurs in somatic or body cells. Those words are pretty much interchangeable. And so those occur in the somatic cells of eukaryotes, which again are multicellular organisms, as a result of asexual reproduction. So in the end, we have two copies of identical genetic information. So if our parent cell has 2N, which essentially means our chromosomes, after mitosis, we will have two daughter cells that also have 2N. So another vocab word is diploid, which essentially means two whole pairs of chromosomes, which would also look like those two X's. So this is just a close-up of some of the review we did. So again, interphase goes into M phase and what mitosis is. So let's start with our first part of mitosis, which is prophase. So our cell is 4N, which are essentially just two copies of chromosomes. And then we have our centrioles, which are on different sides of the cells. And centrioles have spindle fibers that will attach to our chromosomes, which we'll see in a second. And the main thing that happens in prophase is that the nuclear envelope breaks down. So that is a big thing for prophase. And then this is kind of what it looks like. So again, our centrioles have spindle fibers that will attach to our chromosomes. So each one will attach. And on the right, I'm just drawing a more 2D version, which will hopefully be a little neater than yarn and pieces of paper, just so that we can fully visualize that each centriole attaches to each pair of chromosomes. So after that, we have our next phase, which is metaphase. So those centrioles move so that they're on opposite sides of the cell, which are also called opposite poles. And as these centrioles move, because their spindle fibers are attached to the centrioles, spindle fibers also move, which results in our chromosomes being laid straight down the middle. It's also called the equator, if you're really thinking about the centrioles being at opposite poles. That can help visualize it. And again, on my right, just a clearer diagram of what that looks like. And a good way to remember this phase is M is for metaphase, and M is also for middle. So again, during this, we still have our 4N. It's just that they're rearranged in a way that is straight down the middle because our centrioles move. And after this is anaphase. So during this process, our DNA becomes 2N, and the sister chromatins are pulled apart. So these spindle fibers, remember, each centriole attaches to our chromosomes. So these pull apart, and to adjust, the spindle fibers essentially shorten so that there's less connection between the two parts of chromosomes. So again, we went from 4N to 2N. So they're still close to the middle, but now they are very much separated. And our next phase is telophase. So we are still at 2N, but the big thing with this is that the cell is starting to separate. So we can see the two kind of like identical sets of genetic info that is the same as it is in the parent cell and in this daughter cell. And the cell cleavage is going to start. So visually, I will erase down the middle, and the cell is essentially like moving apart so that it breaks into two. And the nuclear envelope that broke down during prophase is now reformed. And the DNA is very much chromatin right now. It's very like loose strands of our chromatin. So this shows how the cell essentially, it looks different. It kind of caves in as it starts to separate. And then I'm just drawing that line down the middle to show that we very much have like two different sets forming, each having same identical DNA. And again, on our right, just another visualization or display of that. We have a wonky looking cell that is starting to spread apart. Two sets of DNA, essentially two copies. So yeah, and then just realizing we don't really need the definition of chromatid here. But anyways, our next phase is cytokinesis. So this is when the cell fully separates. So that cell membrane is being pulled apart, the cytoplasm is dividing, which leaves us with two separate cells that are fully functioning. They have identical genetic information as the parent cell, and it is all about the cell membrane fully separating, and here the cytoplasm dividing, which in the end it looks like this. So two separate cells, identical genetic information as each other, and again as that parent cell. So 2N becomes two cells, each having 2M. And on our right, this is more another way to kind of visualize a separation. So the perfectly circular cell starts to cave in, they slowly separate, and then become two separate circles of cells. So that is the process of mitosis. So now we're going through the functions. What does mitosis do? So mitosis can repair cells. So if there's any damage, then mitosis can help repair it. And it can also help replace cells. So if a cell goes through apoptosis, where it dies, or like anything regarding that, then mitosis can help copy cells, divide them so that there are more. And that happens through asexual reproduction. So one of our functions of mitosis is reproduction. It reproduces cells and helps with growth. So the main thing with mitosis is that it is asexual reproduction. So the DNA from the parent cell is copied so that the offspring is exactly identical to the parent. And this happens in eukaryotes, so multicellular organisms. Now let's go into meiosis. So the phases of meiosis is interphase, which is the same as it is in mitosis. So G1, S phase, G2. Then we have meiosis 1 and meiosis 2. So it goes through 2 rounds of meiosis, which is essentially sexual reproduction, and in the end, it results in 4 daughter cells that contain half the amount of chromosomes as the parent cell. So in our diagram above that, we have the parent cells with 46 chromosomes. So it goes through meiosis 1, which creates 2 cells with 23 chromosomes. Then meiosis 2, which are those blue arrows, leaves us with 4 cells, each having 23 chromosomes as well. So the first part of meiosis 1 looks pretty similar to what it was in mitosis. We have prophase, metaphase, which is prophase we have 4N, metaphase also 4N, and chromosomes lined up down the middle, except it looks different once we get to anaphase, so anaphase 1, which is essentially when instead of the X's kind of being separated, it's out of the 4 X's, 2 X's go to one side, 2 X's go to the other side. And then the DNA uncoils during telophase, and the cell begins to divide, and then it fully divides into 2 separate cells during cytokinesis. So that is meiosis 1. However, the process repeats, so we go into prophase again, but now it's called prophase 2. And instead of having 4 X's, or like our pairs of chromosomes, we have 2. So again, during metaphase 2, lines up down the middle, which brings us into anaphase 2, which is finally when those X's separate. So those pairs are no longer entwined into X's, and we have 4 individual strands, which brings us into telophase 2, which is when the cell starts to divide. So each of those 2 cells start to split in half. So it becomes 4 separate cells during cytokinesis. And then again, just to help show the process, we have our spindle fibers attaching during prophase 1 and metaphase, and then anaphase 1, showing that those chromosomes are being pulled to either side. Goes through telophase, cytokinesis, and then the spindle fibers also attach during prophase 2. Again, now we are in meiosis 2. And then metaphase 2, again, we only have 2 of those X's. Spindle fibers attach during anaphase, which pulls those X pairs apart, pulls the chromosomes apart, which brings us into telophase, where the cell starts to split, and then we get our 4 separate daughter cells. So this is the process of meiosis. So the function of meiosis is to make gametes. So these are our sex cells, so sperm and egg. The main function is to make gametes, and it happens through sexual reproduction. So that is their function, and it occurs in eukaryotes. And the big thing with meiosis is that it creates genetic variation. So I'm just adding in on the mitosis side that there is absolutely no genetic variation at all, whereas meiosis has lots of genetic variation, which occurs from mutations and just the combination of 2 separate organisms' DNA, which happens through sexual reproduction, that will lead to a variety of arrangements of the DNA. And then we have independent assortment and crossing over, so we'll really focus on those last 2. And all of these ways that the organism and offspring can become genetically different happens in eukaryotes specifically, except for the mutations. So let's review what happens in meiosis 1 and meiosis 2. So we went through the process, but what's the big takeaways? So during meiosis 1, the homologous pairs split. So there is a maternal and a paternal copy, and those divide. During meiosis, And that relates to independent assortment because the homologous pairs in meiosis 1 can align randomly, which creates lots of new combinations. So whatever side the maternal DNA goes to or the paternal, it doesn't matter, which creates a number of outcomes. And then crossing over happens when homologous chromosomes that trade the same section of DNA, which essentially allows for that maternal and paternal DNA to happen on like each chromosome. So crossing over is very much how it sounds. So we have the pink, we'll say, is the maternal chromosome, and the green, which is the paternal chromosome. They quite literally overlap, and so that same section, the DNA that the dad has, is going to end up on the mom's chromosome, and the mom's DNA is going to end up on the dad's chromosome. So it is that exchange of chromosomes in that same section of DNA. And then meiosis 2 is when the sister chromatins split, so that copy of maternal and paternal chromosomes split. So again, that's what happens during meiosis 2. And independent assortment and crossing over are ways that the offspring can be genetically different. And again, this is just our overall review. So mitosis is asexual reproduction that happens in body cells and results in two genetically identical daughter cells. Whereas meiosis is sexual reproduction that makes gametes and results in 4 daughter cells that have half the amount of chromosomes as the parent cells. So in meiosis, there's a lot of genetic variation, whereas mitosis, the offspring is identical. And mitosis happens in M phase, so those steps are prophase, metaphase, anaphase, telophase, cytokinesis. And then during meiosis, there is also interphase, meiosis 1, meiosis 2, which leaves us with the outcomes of both mitosis and meiosis. So this is a review. It's helpful for me. I hope it's helpful for whoever's watching as well. Thank you.